Hi everyone! In this lecture we're going to talk about homeostasis. This is the first in a series of lectures to discuss clinical physiology. Grab your coffee, grab some tea, and let's study together. Welcome to Physiology for Students. Today we're going to review the concepts of homeostasis. We're going to define physiology. We're going to look at the general way in which the body is organized, from cells all the way up to the whole organism. We're also going to define the internal environment of the body, intracellular and extracellular fluid compartments. Finally, we'll talk about the ways in which body systems can regulate homeostasis, including negative and positive feedback control mechanisms. So first, let's define physiology. Physiology is the study of the processes within the human body. These are both the chemical and physical processes within the human body. This is different than pathophysiology, which is the study of how these processes can go wrong and cause disease or injury. It's also different from anatomy, which is the study of the structure of the body. So at a microscopic level, the human body is organized from the molecular and cellular level all the way up to the body systems. So we start with the individual molecules, learning the molecular pathways and the biochemistry that underlie the function of the different types of cells within the body. Then we look at the specialized cells and how they come together to create functional units. As the cells create layers of tissues, we then have specialized tissues, for example, connective tissues, epithelial tissues, muscle tissues, and our specialized tissues like nervous tissue. Once these tissues come together, then we have organs. Multiple organs functioning as a unit are defined into the 11 organ systems. It's a little bit arbitrary in a way to talk about organ systems individually, because of course the organ systems are all working together to balance the functions of the body and overall create homeostasis or balance within the body for the whole organism, which is the human being. This organization is similar for animal bodies as well. But for the purpose of this course, we're going to focus on human physiology. Examples of specialized cells coming together to create specialized tissues can be found in the different types of muscle. So look at the images here. You can see, for example, the difference between smooth muscle, cardiac muscle, and skeletal muscle. At a molecular level, there are also differences in the ways that the biochemical reactions occur within each of these specialized cell types. Smooth muscle cells look and function differently than skeletal muscle cells. Skeletal muscle and cardiac muscle have some similarities, but are also different in their cellular structure. Overall, groups of skeletal muscle cells come together to create skeletal muscle, which is like an organ. Groups of cardiac muscle come together to create the layer of muscle within the organ that is the heart. Of course, there are other layers of tissues in the heart, for example, connective tissues as well. And groups of smooth muscle come together to create the smooth muscle layers within the digestive and other organs. When we put this all together, it's very helpful to study both the biochemical, the cellular, and the tissue level functions to help us understand the organs and organ systems. If it's been a while since you've studied physiology, let's review the 11 body systems: the integumentary system, the muscular system, the skeletal system, the nervous system, the cardiovascular system, the respiratory system, immune system, endocrine system, digestive, urinary, and reproductive. There are different approaches to physiology. Usually, we do systems-based physiology, where we look at the individual functions of the different systems and how they create balance within the body. So this balance within the body that we're talking about is homeostasis. Homeostasis is maintaining internal conditions within the body despite external changes. Some really common examples of homeostasis include temperature regulation. Even when it's really cold outside, the internal temperature of our body is within a narrow range, usually near 98 degrees, 98.6 degrees Fahrenheit. We can think of many other ways in which our bodies maintain an internal environment. And usually this is for the biochemical reactions and the health of the cells that need to exist within a certain range of conditions. So homeostasis refers to this balance of the internal environment. What exactly do we mean by the internal environment? When we talk about the internal environment, we're really looking at the fluid and cellular environment within the body. So we're thinking about things like fluid pressure, fluid volume, the composition of fluid, the levels of nutrients and ions, etc. This is where it becomes very important to look at the composition of intracellular and extracellular fluids. So about 60% of our body is water. That's the total body water, or TBW. The water, or the fluid within the cells, is intracellular fluid. The fluid outside of the cells, bathing the cells within the body, this is still internal within the body, is the extracellular fluid. And we can break down the extracellular fluid into the fluid that is bathing the cells between the cells, which is interstitial or between fluid, or the fluid that is within the plasma or the circulating volume. Often we're looking at things like blood pressure and circulating volume. And so this portion of extracellular fluid becomes very important in our measurements. We're going to have a whole lecture on body fluids, total body water, and how to do all these calculations. So stay tuned for that. When we think about fluids and cellular compartments, it's important to remember our membrane biology. So remember that fluids, ions, nutrients, molecules can only move across the compartments of cells if the semi-permeable membranes allow it. So cell membranes separate the inside of the cell from the outside of the cell, or the ICF, intracellular fluid, from the extracellular fluid. So it's going to be a minute for you to remember the channels, the different transporters, and the membrane properties that are going to help you understand the dynamics of cellular physiology. We're also going to remind you of those as we go through the different functions of the cells. So as you're thinking and moving into more clinically oriented physiology, you want to start thinking, how might I measure some of these values if I'm interested in a particular function for a patient or a clinical application? You're going to want to start thinking about laboratory values. What are the normal ranges for ions like sodium, potassium, calcium? What's a normal blood pressure? What's a normal amount of blood volume? What are our normal levels of oxygen and carbon dioxide and nutrients within the blood, that internal environment, that extracellular fluid compartment? So keep in mind that as we're thinking about homeostasis, this is no longer just a physical principle of balancing the body. We're now going to be learning about normal ranges of all of these molecules of interest within the body. So keep in mind your lab values as you continue through this course. Remember that ions, nutrients, wastes, and other substances are regulated to stay within a particular range within the body. And that's why we have lab value ranges, which are within a normal range, or low or high based on particular conditions. When we see that values are outside of the normal range, you want to start asking questions. So pathology is the study of disease or injury. And this is when there are disease or injury processes that are moving the body away from homeostasis. So disease can be considered a disruption of homeostasis. Sometimes it's a response to a disruption of homeostasis. So you want to start asking questions. If you see something in your patient lab values or something in your history and physical exam that is outside of the normal expected range for a particular patient, you want to start thinking, why might this be not within the normal range? What processes are occurring? What could possibly be happening to make this off balance? Is it a compensation because of some change somewhere else? Is it the center of the change or the cause of the change? Or is it secondary to some other change? What would be expected if you can start tracking the processes that you think may be going on? And this is where looking at the molecular, cellular physiology helps us understand what's going on in a particular disease process. And that's where we link pathology to physiology and start studying pathophysiology. So on to the systems of control. What does the body do in order to control ions, nutrients, molecules, various things within the internal environment of the body to keep the body balanced? We have two main types of control: negative feedback and positive feedback. Negative feedback is by far the most common. Negative feedback we consider to be a balancing type feedback control system. So in negative feedback, this is where the body senses a stimulus that is outside of the normal or desired range for the body. And then it creates a signal or a response in the opposite direction to create that balance. So if you have a normal range, let's say for a particular molecule, and the body senses that that molecule is too low, it's going to create a change in the opposite direction. So if that sensor is low, then it's going to move it back up to bring balance to the system. Because it moves in the opposite direction of the stimulus, it's considered a balancing or a negative feedback force. This maintains molecules or set points within a given range. So when you're thinking about negative feedback, you want to ask yourself the main stimulus, the main receptor or sensor, the main effector, or what's going to cause change if something is sensed to be out of range, and the response that that effector will create. I'll give you an example. One really good example of negative feedback is blood glucose regulation. Insulin and glucagon are responsible for blood glucose regulation. So let's think about what happens when you eat a large meal full of sugar. Lots to digest and lots of glucose has just entered the body. So we have a high glucose meal. All of that blood sugar is going to travel to the pancreas. the pancreas is going to sense that blood sugar has increased. What's it going to do? It's going to release insulin. And it's going to send insulin into the bloodstream. Insulin is going to trigger cells within the body to activate receptors for glucose. That glucose is then going to be taken up into the cells and stored. That increased uptake of glucose then causes a decrease of blood glucose. That overall then balances the initially high blood glucose. So when you see negative feedback, what you want to see is that if you have an initially high stimulus, we'll just write a big positive here for a high blood glucose. You see a balancing response, which is in the opposite direction. So the balancing response created by insulin was to decrease blood glucose or to cause a negative decline in blood glucose. That is, the glucose left the blood and went into the cells. That lowered blood sugar, and that was the ultimate response created by the effector, which was the pancreas and insulin. So, pause for a minute. Can you find the stimulus, the sensor, the effector, and the response in this system? Pause for a second and we'll go through that. Could you do it? What was the stimulus? The stimulus in this case was an increase in blood glucose. The sensor was the blood glucose sensors in the pancreas. I know I'm being fairly general about that. We'll get more into these mechanisms in a moment. Don't fault me right now for not getting into the weeds. What's the effector? The effector in this case is also the pancreas, but specifically the insulin secreting cells of the pancreas. Those are the beta cells. We'll learn about those later. What is the response? The response is to decrease blood glucose. It decreases blood glucose by causing uptake of glucose into the body cells. Notice how an initially high stimulus created a stimulus to lower that molecule in the blood. That's a classic negative feedback regulation, keeping blood glucose levels within range. Of course, we know a really common example of when this particular type of homeostasis is disrupted, and that's diabetes. Classically, very high blood glucose levels in diabetes because of the lack of ability to balance. The reasons for diabetes we'll get to later. Okay, positive feedback control. Positive feedback control is more rare in the body. The reason it's more rare in the body is because it creates a domino effect where the stimulus gets bigger and bigger and bigger and bigger. That's not stabilizing. That can create huge changes in the body, and it's very disruptive if it's not expected or not needed. So classic examples of positive feedback control are domino type mechanisms, One classic example of positive feedback control is childbirth. So during childbirth, the baby begins to push on the uterus. Oxytocin levels then increase, sensing the pressure in the uterus. Oxytocin causes contraction of the uterus, causing more pushing of the baby onto the uterus. More pushing of the baby onto the uterus, causing more contraction. More contraction, causing more pushing of the baby onto the uterus. This continues and continues. The stimulus gets bigger and bigger, and the only way it ends is when the baby is pushed out of the uterus for childbirth. So that's a big stimulus that gets bigger and bigger and bigger until the actual initial stimulus is removed. See if you can think of some other positive feedback mechanisms. That's it for homeostasis. Let me know if you have any questions. Further reading can be found in the Guyton and Hall textbook, Chapter 1. If you have any questions, please put them down in the comments. Send me questions anytime. And if you'd like to stick around, we're going to do some challenge questions to test your knowledge.